let's eat a pizza. We'll follow the pizza down the digestive tract as it gets digested and absorbed. Here, the green stuff are the veggies. The white is the cheese. The red dots are the pepperoni. And let's see what's inside each component. The dough of the pizza contains carbohydrates. The pepperoni contains proteins and nucleic acids. The cheese contains proteins and lipids. And the veggies contain carbohydrates and fiber. So let's follow this pizza as it gets digested and absorbed along the digestive tract. So let's begin with the oral cavity. You put the pizza in your mouth and the pizza is digested both mechanically and chemically. Mechanical digestion is brought about by the action of your teeth and chewing. Chemical digestion is brought about by saliva. Saliva contains an enzyme called salivary amylase. In the mouth, when you chew your food, polysaccharides are converted to disaccharides by the action of salivary amylase. Once the food is chewed, it is swallowed in the form of a bolus. A bolus is a mouthful of food that is swallowed. Once it's swallowed and passes through the esophagus, it reaches the stomach. In the stomach, the protein digestion begins. There's mechanical digestion of food by the churning action of the stomach muscles, and there is chemical digestion brought about by the enzyme pepsin. Pepsin breaks down proteins into polypeptide chains. Pepsin is first produced in an inactive form called pepsinogen, which becomes active in the presence of hydrochloric acid. So that's proteins becoming polypeptides, and it's brought about by pepsin. Hydrochloric acid not only converts pepsinogen to pepsin, but it also destroys any bacteria that might be ingested along with the food. Food that leaves the stomach is called acidic chyme, and it enters the duodenum, which is the first part of the small intestine. In the duodenum, the gallbladder releases bile, which emulsifies the fats, allowing them to mix with water. The pancreas release a whole host of enzymes into the duodenum so that they can act on the acidic chyme and break all 4 macromolecules down. Polysaccharides are converted to disaccharides in the presence of pancreatic amylase. Disaccharides are not monomers, and so they are not ready for absorption yet. Nucleic acids, on the other hand, are broken down into nucleotides, which are monomers, and they are ready for absorption. This conversion is brought about by nuclease, also produced by the pancreas. Lipids, which are now emulsified, are broken down into glycerol and fatty acids that are monomers of lipids and ready for absorption. This conversion is brought about by pancreatic lipase. Finally, polypeptides are broken down into oligopeptides, which are made up of a few amino acids strung together, and these are broken down by 2 enzymes, trypsin and chymotrypsin. Oligopeptides are not ready for absorption. So disaccharides are not ready, nucleotides are ready, glycerol and fatty acids are ready, but oligopeptides are not ready for absorption. Now it's time to break down the nutrients in your pizza into its monomers. This happens in the jejunum and ileum of the small intestine. The lining of the jejunum and ileum is made up of cells that have microvilli. These microvilli look like a brush, and so the enzymes produced by these cells are called the enzymes of the brush border. These enzymes have the ability to break down dimers into monomers. So maltose, which is a disaccharide, is converted to glucose and glucose, 2 molecules of glucose, by the enzyme maltase. Lactose, which is a disaccharide, is broken into glucose and galactose with the help of the enzyme lactase. Sucrose is broken down into glucose and fructose with the help of the enzyme sucrase. Glucose, galactose, and fructose are monomers and ready for absorption. Oligopeptides must be now converted into individual amino acids. This is brought about by the enzymes carboxypeptidase and aminopeptidase. Amino acids are monomers and therefore ready for absorption. So all the macromolecules in your pizza have been broken down and ready for absorption. In order for absorption to take place, molecules must move from the intestine to the capillary. The capillary is fenestrated, which means it has holes in it, allowing molecules to move in fairly easily. Molecules must move from the intestinal lumen, which is 1, to the intestinal cell, must enter the intestinal cell, and then leave the intestinal cell and enter the interstitial fluid or space. From the interstitial space, the molecules must enter the capillary, which is a fenestrated capillary. Absorption of nutrients takes place in the small intestine. The molecules must move from the lumen to the intestinal wall, to the interstitial space, into the capillary. The carbohydrates that must make this journey are glucose, fructose, and galactose. So we are moving from the lumen, entering the intestinal cell, leaving the intestinal cell, and entering the capillary. From 1 to 2, glucose, fructose, and galactose use secondary active transport, and they are moving along with sodium. From 2 to 3, they travel via facilitated diffusion. From 3 to 4, entering the capillary is done by simple diffusion. The absorption of proteins is very similar to the absorption of carbohydrates. In this case, we're talking about the movement of amino acids. Amino acids move from 1 to 2 via secondary active transport, 2 to 3 via facilitated diffusion, and 3 to 4 via simple diffusion. And now for the absorption of lipids. By this time, the lipids are in the form of fatty acids and glycerol. We must remember that fatty acids and glycerol are hydrophobic molecules, and therefore they can cross the phospholipid bilayer. This allows them to very easily enter the intestinal cell via simple diffusion. So that's them entering the intestinal cell. Once they enter the intestinal cell, the fatty acids and glycerol reform into triglycerides and become covered by a protein coat. Together, these little packets are called chylomicrons. Chylomicrons are too big to leave the cell via facilitated diffusion, so they use exocytosis in order to leave the cell. They enter the capillary via simple diffusion. At this point, it is important to note that the capillary that we are talking about are lacteals or lymphatic capillaries and not blood capillaries.